Hi everyone, my name is Marcus Gray. Thanks a lot for the invitation to speak here at QAAFI, and thanks to Eugenie Rory for inviting me as well. We're doing some research, some collaborative research together. It's been led by Mr. Peng Fei Han in the audience. I probably won't talk much about that project yet, but we're expecting some fairly exciting results soon. So I work a lot with brain imaging. You can see a nice image of the brain here, or the central nervous system. But an area that's sometimes neglected is the peripheral nervous system, which you can see illustrated here. These are the nerves that connect the body to the brain, and within this system you've got the autonomic nervous system, which regulates how bodily responses and organs within the body function. This talk's called Predicting the Body, and it's about how the autonomic nervous system integrates information about the body during emotion. So you probably all here would be quite happy to accept the statement that the mind is fundamentally embodied. Despite any sort of religious or dualist beliefs that we might have, I think most of us would accept that the mind arises from the brain, and if you damage the brain or the body, it's going to have an influence on the mind. My research likes to go a bit further and suggest that actually a lot of mental activity, including cognition, emotions, and executive processing, interacts quite subtly with the control of internal physiological state. And it can be understood as arising from homeostatic emotions. So homeostatic emotions are things like thirst, hunger for air or hunger for food, pain, sensitivity to cold, sexual arousal, these sorts of things. And homeostatic emotions have two sort of principal defining features. They're really strong motivators of behaviour, and they're relative. So if you think about cold water and the appeal of having a glass of cold water when your body's really hot is quite different to if you're hypothermic or extremely cold. The emotional content is quite different. And it's probably likely that homeostatic emotions arose during evolution as an effective way of ensuring survival. So if you're motivated to keep your body in good shape and eat and drink when you need to, keep warm, you're going to have a better chance of surviving. And it's likely as humans evolved in social settings that these systems became more sophisticated and might have led to more complex emotions, things like pride or shame, self-esteem. These are all quite important in being successful in social environments and have consequences for longevity and mating and survival as well. So we think possibly emotions may have evolved from homeostatic systems earlier in evolution. So this creates a sort of dynamic system, the self, which is constantly integrating and communicating with the body within an environment. Today I'd just like to talk about a few things, some theories about relationships between the body and the brain, before talking a bit about interoception or the sensing of the body, and then I'll talk about some research I've done in the cardiovascular context, before talking briefly about inflammation and how that affects mood and cognition, and how individual differences are important in these systems. And finally, I'll just mention in passing, in conclusion, the sort of new emerging framework that emotion theorists are starting to adopt, which sees emotion in terms of hierarchical predictive coding and error prediction. So we've long recognised that there are close links between the body and emotions. Hippocrates actually proposes a fairly modern view here when he says that the source of our pleasure, merriment, laughter and amusement, grief, anxiety, etc. is the brain. This enables us to think, to see, hear and distinguish between the ugly and the beautiful, the bad and the good, pleasant and unpleasant, and he specifically states that neither the diaphragm nor the heart take part in mental operations. It's interesting that this view wasn't particularly popular. Aristotle's view was much more widely accepted. A little bit later he said the brain is not responsible for any of the sensations. The correct view is the seat and source of sensations is the region of the heart. the emotions of pleasure and pain are generally all sensations plainly have their source in the heart. Well, Aristotle was clearly wrong, but it does demonstrate, I think, quite clearly the recognition, the long recognition we've had that the body is important to emotion and involved closely with emotion. If we look at some more modern theorists of emotion, we've got James and Langer here. They were probably the first in the modern era to theorise about emotion, and they're famous for proposing the James-Lange hypothesis. Basically, they highlight the importance of the body in emotion, and a nice example that's often mentioned in psychology courses is James's example of the bear. So he asks you to imagine that you're walking through the forest and you see a bear. A short time later, you find yourself scared and running away. And we would naturally think that the logical sequence is you see the bear, you become scared, and then you run away. But James Lange sort of reversed this order and said, actually, in fact, what happens is you'll see the bear, you'll start to run away, and after that you'll become aware of your arousal responses and you'll interpret that as fear. So he sort of reverses the role of the body in emotional generation. So he sort of summarises this quite nicely. I guess it's probably summarised well here by saying that we feel sorry because we cry, angry because we strike, afraid because we tremble, and not that we cry, strike or tremble because we are sorry, angry or fearful. Without the bodily states following on the perception, the latter would be purely cognitive in form, pale, colourless and destitute of emotional warmth. So we might then see the bear and judge it best to run, receive the insult and deem it right to strike, but we could not actually feel afraid or angry. So he says that the feelings of these emotions is the perception of the bodily response. That was a really influential theory and had a big impact in psychology. It was critiqued quite extensively by Cannon and Bard, and they had 5 basic points what they thought was wrong with this theory. The first one is that if you separate the viscera from the nervous system, it doesn't actually change emotional responses or behaviours in animals. But that slightly misses the point. What we're talking about here is theories of emotion, perceived emotion, not emotional responses. And you can't really directly assess the emotion experienced by an animal. The other ones have more merit, perhaps. They say the same visceral changes occur in different emotional states, and the viscera is relatively insensitive. So the viscera is the organs within your body, like the heart, the lungs, the diaphragm, the stomach, the bowel. They would argue that they're not sensitive enough to have a unique signature for each different type of emotion. And there is some merit to that, although recent research is demonstrating there is more subtlety in these systems and the coordination between these systems than perhaps Cannon and Bard realised. The next major theorists I'd like to highlight are Schacter and Singer here, and they proposed a well-known 2-stage theory of emotion. Basically, they drew on some previous research which had been developed around the 20s, where experimenters injected people with adrenaline and then looked at emotional responses. What they did, they did this covertly. So in one of their famous experiments, they got volunteers to come in and told them they were looking at a study on the influence of new drug, Suproxen, on visual processing. This was a ruse, however. They injected them with adrenaline and then they asked them to wait in a waiting room, and they were paired with a confederate that was either really annoyed and agitated, frustrated, muttering and swearing, or someone who was extremely elated and happy, giggling, laughing. After about 15 minutes, they got the subjects in and assessed their emotions, and they found that the people who were paired with the agitated or negative confederate reported negative feelings, whereas the people that were paired with a sort of more positive person reported feelings of elation and joy. And that's interesting because it highlights that it's not simply the perception of bodily arousal in this case that leads directly to emotion. There's also an important influence of interpretation. So we interpret the state of our bodies in a way that's consistent with our environment, and in a sense we're making predictions about how our environment may have caused our current bodily state, and when we find a compelling explanation for that, we will experience the associated emotion like agitation or excitation. Antonio Damasio down here also made some fairly significant contributions to emotion more recently. He published work in the 90s about the somatic marker hypothesis, which essentially says that our body creates somatic signs or signals that we can use as cognitive shortcuts. So he highlighted a fast-track decision-making system that can respond rapidly and advantageously in most situations. A good example of that was demonstrated by gambling tasks. He got people to do the Iowa gambling task where they showed they have to make bets on a deck of cards and have to learn which cards are associated with reward and which cards are associated with loss. And what he found that before people recognised consciously which were the best decks, best cards to pick, they were having autonomic responses to the good and bad decks that predicted their later recognition. And what's interesting is in certain people, including people with psychopathic disorders, they found a very poor learning behaviour that was associated with disturbances in their normal autonomic responses to loss and failure. So that suggests that our body can provide these rapid somatic cues, which we can then use to navigate our way through the environment that influence emotion. So that brings us to Bud Craig, the last figure listed here, and he's probably less a theorist of emotion than a neurophysiologist who was studying the actual circuitry that underlies interoception. So interoception is the process by which we sense and aware of our bodily state. I guess the traditional views of interoception came from people like Sherrington, who sort of described a number of ways we can be aware of our body, things like teloreception, which is vision and hearing, proprioception, so being aware of our limb positions, exteroception, or touch, sense of touch, chemoception, taste and smell, and he placed interoception as the fifth separate system. And there was work sort of highlighting the neural pathways through the senses up through the spinal cord and into the brain for each of these sort of systems. Bud Craig was important because he did a lot of work on the Lamina 1 spinothalamo-cortical pathway, and his work suggests that rather than interoception being understood simply as the perception of signals from the viscera, it should possibly be broadened to describe the sensation of all aspects of the body, including touch, sensual touch, heat, pain. All of these things are transmitted through the Lamina 1 spinothalamo-cortical pathways. He highlighted a few unique pathways that we have in humans that are not there in primates, and he suggests that direct projections from brainstem regions, including the nucleus tractus solitarius up to areas of the thalamus, provide humans with a unique way to be aware of their body and a unique pathway for the body state to influence emotion and behaviour. He was able to trace these pathways up to cortical regions, including, importantly, the anterior insula, which is sort of behind where your ears would be in the brain. And it's quite a high-level cortical region that you wouldn't generally associate with lower-level bodily processing. And it makes a lot of sense when you look at some of the imaging studies about different types of body sensations. We see activations in the insula here for things like graded cooling, C-fibre or light sensual touch, as well as thermal pain or chronic pain. There's a whole range of manipulations to the body that will cause activations in these higher cortical regions, including the insula. It's interesting that you also see the insula light up to a lot of emotional stimuli. So anger, the subjective feeling of coolness, the perception of disgust or trustworthiness in facial expressions of others, or sexual arousal, they'll all also activate this region of the anterior insula. And Bud Craig was one of the first to suggest that this region is important for integrating cognitive, emotional consciousness with the signals coming from the body. So that suggests we do have a fairly unique and evolved way to sense our bodies, and that's integrated with ongoing emotional processing. What happens when the system is compromised? A nice example comes from this paper that was published in Science in 2007, where they looked at smoking behaviour in people that had strokes, different lesions to parts of their brain. And they divided up their patients into people that had strokes within the insula or people that had strokes in regions that didn't include the insula. So you've got 19 insular lesions and 50 lesions elsewhere in the brain. And they got these people and they assessed smoking quite comprehensively after their lesion, and in the weeks following they asked how much did you used to smoke, how much do you feel like smoking now. In people that quit, they sort of said how difficult was it to quit, how much of an urge did you have to stop smoking, did you have any relapses, these sorts of questions. And what they found was the location of the lesion didn't in itself predict quitting, but in the people that did quit, there was 14 people who quit with no particular disruption of their smoking addiction. So essentially, people that had lesions in the insula and stopped smoking were much more likely to find it much more easy to stop smoking than people that didn't. So yeah, they were twice as likely to have suffered insular damage if they were able to effectively quit smoking. That was a really intriguing finding, but NACFI didn't go terribly far in explaining why this might be the case. We were asked, my previous boss and myself were asked to write a review or a commentary on these findings in Neuron, which we did, and we sort of introduced the idea that interoception is probably the system that mediates these effects. We know a lot about addiction in terms of neurochemical pathways, dopamine in the brain, these sorts of things, but the subjective urges and feelings of cravings are also an important feature of addiction, and they're important in mediating relapse or success in quitting. And we highlighted that the insula provides the capacity to represent and respond to the internal stimuli so that they may be perceived subjectively as anxiety or tension or craving states. And we highlighted Budd-Craig's pathways and suggested that this system is probably the reason why these people that had lesions in the insula found it much more easy to stop smoking and didn't particularly go through craving. A lot of my work in the UK was sort of done in a cardiovascular context. So I want to highlight a few studies now that have looked at the heart in terms of relationships between the body and the brain. In particular, I want to focus on three basic studies: responses to pain, and then the motivational salience of painful stimuli, and finally on conditioned fear and extinction. So we know that cardiac function is regulated primarily by brainstem nuclei. Within the pons and the medulla, you've got some fairly simple 2 or 3 synapse systems that regulate reflexes that keep the heart running normally. But we also know that emotional and cognitive stresses can have a quite profound influence on cardiac function. So if you get someone in a stress state, you'll see stress hormone responses, an inflammatory cascade, including circulating catecholamines, which themselves can induce changes in cardiac function, changes in the contractility of the heart, the speed of heart rate, and the evenness of repolarization within the heart walls. But you've also got direct neural pathways that mediate these influences. But what's interesting is over and above these sort of lower level brainstem, medullary and pons systems, we've also got a hierarchy that includes higher regions of the insula, cingulate and amygdala. And these are limbic regions, or regions that are involved in generating emotional responses. The primary source of signals from the heart, I guess, comes from what's called baroreceptors. And they're pressure and stretch sensitive receptors, and they're located primarily within the aortic arch and the carotid body veins. And so each time the heart beats, it squeezes out a bolus of blood which distends the walls of the heart and the aorta and the great veins around the heart, the great arteries. That activates baroreceptors, which transmit up into primary regions of the brainstem. And the baroreflex itself regulates heart function on a sort of beat-by-beat basis. So if your blood pressure increases, the baroreflex will act to decrease your heart rate and increase vasodilation peripherally. So that will act to reduce blood pressure. Conversely, if your blood pressure is low, it will speed up your heart rate and cause vasoconstriction, which helps maintain your blood pressure within a certain band that's useful. There's other inputs that also integrate with the baroreflex, including chemosensing, the renin-angiotensin system, cytokines, which mediate inflammatory responses. And as we said, there's descending influences from higher limbic cortical regions. And the primary outputs are through the sympathetic and the parasympathetic nervous system. But what we're interested in is, could we see higher control directly in a neuroimaging study that demonstrates higher influences generated from cardiac afferent signals, so signals from the heart? Do they just stay in the brainstem regions, or do they also propagate higher in the nervous system? We know that, for example, in the insular cortex, there are individual neurons that respond to changes in blood pressure. So if you give someone an injection of phenylephrine, that will increase blood pressure, or sodium nitroprussate will decrease blood pressure. And there are individual neurons that in the insular cortex that will stop firing when blood pressure increases or start firing when it drops. And likewise, there are neurons that will start firing when blood pressure increases and decrease when it drops. So that's sort of showing a sensitivity within these higher cortical regions to fairly general changes in blood pressure. What we're more interested in is the sort of moment-by-moment signals from the heart. Do they also have an influence? So we studied this with a functional MRI study. We got 11 healthy males, and we put them in an MRI scanner. At the same time, we measured a whole range of body responses, including skin conductance, continuous blood pressure responses, pulse oximetry and ECG. And we gave them a number of painful and surprising electric shocks to the back of the hand. An important thing is we timed the shocks to be delivered at different periods of the cardiac cycle, so either synchronous with the R wave or delayed. We know that baroreceptors are generated by blood being ejected from the heart, and that's happening in this period here. The T wave represents the end of the cardiac contraction when baroreceptors are firing their most, whereas a synchronous period, they're generally silent. So by delivering shocks at different periods of the cardiac cycle, we're able to look at the influence of these momentary signals from baroreceptors up into the brain. We saw quite clear differences in blood pressure responses. So when we gave shocks to people, these are cardiac cycles after the shock. They had a very consistent and clear increase in blood pressure for about 10 beats after the shock. That was when we gave shocks when there was no simultaneous baroreceptor activation. But when we gave the shocks during baroreceptor activation, the responses were markedly attenuated. And that makes a lot of sense when you think about the sympathetic nerves in the body. If you record from these nerves in the periphery, you'll see they have a rhythmicity that's directly linked to the heart. So when the heart fires, it inhibits sympathetic outflow to the body, and it's also inhibiting cardiovascular responses to pain. We saw similar patterns in systolic and diastolic blood pressure. Previously, that was mean arterial pressure. We also looked at the neural correlates, so what was happening in the brain that reflected these differences in blood pressure responses. And we saw differential responses within the amygdala, within the insula, and within primary brainstem autonomic nuclei. So within the insula and the brainstem, we see a reduced response associated with the reduced blood pressure response. And conversely, in the amygdala, we saw an increased response when this blood pressure response was inhibited. We also looked at parasympathetic nervous system activity. One way you can measure the activity of the parasympathetic nervous system is to look at heart rate variability. So these are the heart rate period over a number of beats. This is one subject here in red and another subject here in blue. And what I'll point out here is the sort of smooth nature of changes in heart rate in this subject versus the very rapid changes in heart rate in this subject. So this person has much greater variability in the timing of their heartbeats, and that's a very good thing. That's a good index of parasympathetic function, and parasympathetic system control of the heart is good for keeping your heart in good shape, dealing with stresses and environmental stimuli. It's protective against stroke and heart attacks, and it's generally considered a good thing. When we looked at differences between people with high heart rate variability versus low heart rate variability, we saw that that also mapped out very nicely to the differences in insular function between these two periods of shock. So the people that responded most differently to these two shocks also had the greatest level of parasympathetic activity generally, and we see a similar thing in the very acute changes to the shock. So after each shock, the change in the heart rate variability was also reflected by difference in brain response in the brainstem and in the amygdala. So that shows us quite clearly that these moment-by-moment signals from the heart are transmitted up to fairly high regions of the brain. They're doing more, than just regulating brainstem responses. They're also influencing function in higher brain regions, regions that we know are important with emotion and cognition. And that's an interesting finding, I think. And it sort of motivated the next study. We sort of thought, well, if we get different responses in these higher cognitive regions and emotional regions, depending on the signal of the heart, could that also result in different emotion perception or emotional behaviour? So we thought we'd look at that with another study into pain and baroreceptors. There's a few things about pain that we already know. That pain responses are linked to blood pressure in a surprising way. Essentially, increased blood pressure reduces your sensitivity to pain. If you get animals and you train them to run on a treadmill, for example, to avoid painful stimuli, they'll do that quite happily and they'll work very hard to avoid painful shocks. But if you then remove their parasympathetic nervous system, they become much more blasé and they don't work very hard to avoid shocks. You've also got higher pain thresholds among essential hypertension patients. So if you're going to the dentist and you're getting your teeth drilled, people with higher blood pressure generally have a higher pain threshold as well. So it suggests there's an association between pain and blood pressure. We also know that attention can influence pain perception. So if you're focusing on a painful stimulus, you'll generally find it more painful than if you're distracted. And there's a large number of experimental studies which have given people things like addition tasks during pain stimuli versus asking them to focus and anticipate the pain. And not only is the subjective experience of pain greater when you're focusing on it, your neural response to pain is also increased. So it's not purely just a subjective thing. Your central nervous system will represent the pain more if you're focusing on it. So we were interested in the question, might there be an interaction between attentional focus and pain? We did this in a probabilistic cue paradigm. So we got people and we measured EEG and we showed them a series of simple shapes on the computer screen. Some of these shapes were paired with shocks and some weren't paired with shocks, about 50% of the time. So after a while, people could tell that this circle was followed by a shock most of the time, whereas this triangle was never followed by a shock. And again, we gave the shock stimulus at different times of the cardiac cycle, so when baroreceptors were active versus when they were silent. These are the results of the ERP data we saw. So if you get a sensory stimulus, you get a characteristic ERP waveform with a negative component and a positive component. And this positive component is also sensitive to things like subjective association or perception, whereas the N2 component much more basically reflects the stimulus intensity. And we saw an interaction in the P2 amplitude depending on what period of the heart the shocks were delivered in and whether they were expected or not. In order to explain that interaction a bit more, we did some post hoc ANOVAs. So during the first component, the N2 component, there were no significant effects. But during the P2 component, when we looked just at the cued stimuli, we saw there was a difference between active versus silent periods of the heart. And conversely, when we looked at just the silent periods of the heart, we saw there was a strong difference between cued and uncued pain. And we sort of summarise that with this model, which suggests how baroreceptors and attention might be related. Basically, if you get a pain stimulus, that will produce a pain evoked potential in the cortex, and attention increases that. So if you're attending it, that opens the gate, you get a larger P2 component. Baroreceptors appear to modulate this gate. So if baroreceptors are active, they will close the attentional gate and abolish the effects of attention. So that suggests really quite a subtle interaction between ongoing cognition and attention and the influence of the heart signals on the processing of pain stimuli. One last study we did along these similar lines, we thought, well, this is interesting. You know, we've got an interaction with attention, which is a high-level cognitive process. What about things like fear conditioning? We know that we can condition fear in the laboratory. For example, if you present someone with a red circle or a green circle and you pair that with an aversive stimulus like a burst of loud white noise, you'll come to associate the stimulus with this unpleasant stimuli. Conversely, we presented a blue circle which was never paired with unpleasant stimuli. So we conditioned the red and the blue stimuli negatively, and we didn't condition this blue stimuli here. Extinctions are a common way to study fear conditioning. Once you've established conditioned fear, the stimuli themselves will tend to evoke negative emotions and fear. But this will habituate if you carry on presenting the stimuli with no negative associations. So if you then present the conditioned stimuli over and over again and don't pair them with unpleasant stimuli, the fear decreases, it extinguishes. These processes are important for things like post-traumatic stress disorder. We know there's significant or specific deficits in this extinction process in certain psychiatric conditions. So what we did here is we got subjects, and on the first day we set up the fear conditioning, and we delivered all the stimuli when baroreceptors were inactive. We then later in the day tested them again during extinction, but we paired the green stimuli with the active condition, baroreceptors active, and the red stimuli when the baroreceptors were silent. We then got them back on the second day and looked at reinstatement. One of the things that will happen with conditioned fear is after it's extinguished, you might go home, the next day you might see the stimulus again and you'll get a reinstatement. The fear responses will come back again, and eventually they will extinguish again as well. So we also looked at reinstatement on a following day for these three types of stimuli. What we found was during the extinction period there was significant differences in heart rate response to stimuli that were extinguished when baroreceptors were active versus when they were inactive. So you get a much greater heart rate response during extinction when you're presenting stimuli when the baroreceptors are silent. If you're extinguishing when the baroreceptors are active, you get a potentiation of extinction. Extinction happens more easily. And the same thing happens in reinstatement. These previously extinguished stimuli were reinstated more if they were reinstated when the baroreceptors were inactive. So these signals from the heart have an influence on extinction, and that is also reflected in reinstatement the next day. We saw corresponding neural activations in the amygdala and in the thalamus, so that when extinction was potentiated by baroreceptors, we saw increased activity in the amygdala and in the thalamus. So that suggests again that there's a really quite a complex and subtle relationship between ongoing physiological signals, in this case from the heart, and emotional or cognitive processing, things like fear. I want to also briefly talk about some work we've done with inflammation. Inflammation's a really interesting area at the moment, and I've got some other collaborative work with people who are looking at the gut-brain axis and how inflammatory activity within the gastrointestinal system influences mood and cognition. A good way to think about this is to think about sickness behaviour. So we all know what happens when we feel sick. but probably not many of us realise that it's a stereotyped behavioural pattern that we experience when we're sick. Things like reduced motivation, psychosomatic, psychomotor slowing, fever, and alterations in cognition and affect are constantly paired with inflammation. We know that these are mediated by pro-inflammatory cytokines. So if you denervate the autonomic nervous system, you don't get these behavioural stereotyped patterns with inflammation. Also, we can see early gene CFOS activation when cytokines bind to the peripheral receptors in the autonomic nervous system, and that is associated with direct activation in neural structures, so that we know that peripheral cytokines directly activate the brain structures. And the regions they activate are implicated in homeostasis and emotion, the same sort of regions we've already seen. So we wanted to look at, well, what are the neural origins of sickness behaviours? So we did this study in 10 healthy male participants, or 16, sorry, and we looked at that during a simple cognitive task. We induced inflammation by injecting the subjects with typhoid vaccine. We also injected them with a saline in a randomised placebo-blind crossover design. And the task was a simple colour stroop task. So it's got two conditions here, and what the subject is asked to do is to, with a button box, identify the colour of the ink of the top word. So in this condition it's quite simple. The correct response is red because the ink here is red. The incongruent condition's more challenging. The ink is again red, so the correct response again is red. That's more difficult generally. People are a bit slower, they find it more demanding, and it's a good cognitive activation task. So we took a number of measurements during this study, including plasma interleukin-6 and tumour necrosis factor, as well as cortisol, temperature, and measurements of mood. We saw, as we would expect, significant increases in peripheral interleukin-6. It's a pro-inflammatory cytokine after the injection, much more so than during placebo. We didn't see changes in other cytokines consistent with previous results, and there was no differences in temperature or cortisol that were specific to the inflammatory state. We also saw, as you'd expect, the incongruent condition was a bit harder, they were a bit slower, and they made more errors. But there, again, there was no interaction between inflammation and task condition. What we did see, however, was changes in mood, specifically confusion and fatigue. So after 3 hours, people that were in the inflammatory state reported significantly greater confusion and significantly greater fatigue. We then looked at the neural correlates associated with these behavioural changes. In column A, you'll see the simple activations to the task in regions we'd expect, the inferior frontal gyrus, visual areas, intraparietal circles associated with attention. We also see activations for inflammation, so within the dorsal insula, amygdala, the thalamus, and the periaqueductal grey. But what we also saw was an interaction in neural responses between inflammation and between task state. So, for example, in the dorsolateral prefrontal cortex, you get a very small increase in activity while on placebo, but a much larger increase when you're inflamed. Similarly with the anterior cingulate, a much greater increase in response in the inflammatory state during the harder component. We also looked a bit further and looked at the bold responses that reflected fatigue in this column and confusion here, and we see clear correlations between bold responses in individual subjects and the level of fatigue they reported during inflammation in the insula left and right and in the cingulate, and in confusion in the cingulate and the insula, negative correlations in the left prefrontal and amygdala. So these findings suggest, I think, quite nicely or confirm the ideas that peripheral inflammation selectively influences paralimbic or emotional neural activity to generate the core symptoms of sickness and to reorient the basic motivational state. We also wanted to look at mood changes more specifically. We know that depression's important and it's tied to inflammation in a number of ways. Inflammatory cytokines, they're implicated in the pathophysiology of depression. So if you give someone interferon alpha treatment, for example, that induces clinical depression in about a third of patients. And we know that in depressed patients you see increases in pro-inflammatory cytokines. So there's a link there between these two things that suggests that inflammatory mediators may contribute to the pathophysiology of depression. We looked at that in the same subjects during an emotion processing task. They were simply shown different facial expressions and they were asked to make a simple judgment. Do you think this person is younger or older than 25? It's an irrelevant task that just makes them focus on the faces so we can activate emotional processing circuits. We saw, again, a range of activations associated with visual processing and emotional processing, but I'll highlight here this region, CG25. What we saw here is a direct association between the amount of inflammation-induced mood change, confusion and fatigue, and neural responses in this region. And that's really interesting because this region we know is closely tied to depression. One of the treatments for depression is deep brain stimulation, where you insert electrodes into the brain and stimulate it. And the region they target is this region here, CG25. We also looked at how this region connects to other regions of the brain, and we saw significant changes with regions including the medial prefrontal, superior temporal sulcus, and the nucleus accumbens. So that demonstrates that the people who showed the greatest mood change during inflammation showed the greatest difference in connectivity between these regions of the brain. So that again suggests, I think, quite nicely that inflammation and depression are quite closely linked, and they seem to be linked by the neural structures that are associated with representing the body and processing emotions generally. We know that there's differences between people as well. People are all quite different. The way we respond to emotional stimuli differs, and the way we respond to our own bodies differs between different people. A nice example of that is this study by Hugo Critchley. In this task, he looked at interoceptive sensitivity. How am I going for time? 2 minutes. Okay. In this task, subjects were played a series of notes, and we timed the notes to be either synchronous with their heart or to be faster or slower than their heart rate. There was also a slightly deviant note included that was slightly above or below the tone of the previous notes. So the task was, in one condition, tell us whether it's matching your heart, or in the second condition, tell us if it included a deviant note. So that allows you to focus attention either on interoception processing or on processing of auditory stimuli. And what we saw was increased focus on your body activated quite strongly the anterior insula and a few other regions. And there was an interaction within a network of regions as well during greater increases when the stimuli did not match your body over the note judging tasks. And when you mask these findings by these to look at the interaction neurally in regions that are primarily responsive for representing the body, you'll see the anterior insula come through again. So anterior insular activity increases when the notes are not matching your heart versus when they are, when you're trying to sense your heartbeat, versus when you're judging notes, it reduces when the beats aren't in line with your heart. So that sort of suggests that the anterior insula is doing a sort of comparison. It's trying to sort of sense the signals from your heart, and when you're trying to become aware of them, it's predicting when there's a mismatch between what you're expecting and what you're presented with. What's also interesting is the associations with anxiety. So the functional response is also mapped very closely on their accuracy in heartbeat detection, so how generally aware of their body they were. And that their performance on the task was mapped by anxiety levels, general anxiety reported by the subjects. And the insular activity also reflected anxiety levels. We also saw the same sort of associations with the size and shape of this region. So the volume of this region of the brain was larger in people who could detect their heart rate better, and in 2 regions of the insula. So again, it highlights the close association between the insular cortex and the processing of bodily signals. These were also mapped out by perception awareness. So if you're better at perceiving your body, the size of this region is also increased. So that's all the experimental work I'll cover. 5 minutes? Sure. I'll just finish by highlighting a sort of a new emerging paradigm. And that sort of tries to tie together these new insights in a new way. And it's called the hierarchical predictive coding model. It draws on a lot of other research, which I'll only cover very briefly here, but research about prediction error signals and how the brain might work as an error prediction machine. There's good examples of how the visual system might work to predict stimuli. This sort of typical Necker cube illusion is typically seen as either this shape or this shape, and it sort of flips between these two. And we think that's explained by the fact that you have a high-level prediction about the cause of this sensory stimuli, that it's either this or this, and your perception will flip based on the evidence for each model at a particular point in time. This framework draws on earlier work by people like Helmholtz, who said that perception is a method of prediction testing. So you perceive the world in order to test predictions about the world, and specifically predictions about the causes of your sensory stimuli. That's been integrated with work about the free energy principle and sort of new progress in statistical modelling. And it's been formulated in a hierarchical model that matches out what we know about brain structure, that information is passed through a hierarchy of neural structures, and in these structures predictions are passed from high levels down to low sensory levels, and errors are passed in the opposite direction from low levels up. So we might have a prediction. If I'm standing here giving this talk, I would assume that I'm sort of standing upright, so I would have a prediction that the sensory information from my vestibular system and pressure from my feet should have a certain shape, and as long as that prediction is confirmed, it's all good. If there's an error prediction, then that will cascade up the hierarchy and cause me to really re-evaluate my overall belief. I won't bother with any of that, but just to finish with, emotion is sort of being understood within this framework as well now, and I think this is a really interesting area for new research. It suggests that emotion is actually a process of interoceptive inference. So we have a brain that receives sensations, that produces internal states, that leads us to act on the world, which changes our external states, and these loops are continual. What we're doing is we're predicting the current state of our body and why we think we should have the current sensory input that we do. And by modelling this in a hierarchical prediction model, we're hoping to make advances in the way we understand how the brain processes these signals and how they're integrated with emotion and consciousness. So that was a fairly rapid talk. I'd just like to acknowledge a lot of this work was carried out at the Wellcome Trust when I was there in London. Professor Hugo Critchley's lab with Neil Harrison and now Sarah Garfinkel and Anil Seth's lab. There's important theoretical developments from Carl Friston, Ray Dolan, Andrew Steptoe and a few others. And currently we're doing some collaborative research with Eugenie and Peng-Fei Han to look at how taste is instantiated within these systems. Thanks.